Okay. Hi guys, welcome. I'm just setting up over on my YouTube channel. So please do come along if you are wanting to join me. If you are just on here for the TikTok side of things, then please ask questions away here. I will jump between looking there and what's going on the screen. So yeah, welcome if you are just joining me and you are interested in A-level biology OCR A. If we have any questions, the exams are getting so close now, it's a bit scary how close they're getting for you guys. Andreas, hi. Hi, welcome. Welcome. So if you have any topic areas you want me to cover, please do ask away. I'm happy to start the ball rolling with any topic area, or if you have a specific question. So Andrea, since you're the first one and only one in here at the moment, is there anything you are interested in covering? So guys, if you want to, please do come join me on my YouTube channel. And also, if you have specific topic areas you would like me to cover, then please do ask away. Great. Which exam paper have you been doing and which question? What year is it? Do you know? But if you have a question you want to ask, please do come and ask it. If you are on here and you're interested, paper 1, 2020. Right, let's have a look. Which question on 2020? I will have a look at that one specifically for you. Paper 1, 2020. Question 17C. Right, let me have a look. 17C. Hi guys. Oh, thank you for the roses. Right, 17C. Let's have a quick look. That's 17C. Composite 18. 17. So it's a TLC one. Okay. Let me just bring some stuff across. I'm bring this down here right away. Okay, so let's have a look at this one then. So 17. Oh. Okay, so this question then is looking at the confused by RUBP concentration decreasing after reduced carbon dioxide. Why is it decreasing and not keeping constant? Okay, so you're being told that you've got this photosynthetic pigment. You've been told about the practical that you can do to do TLC. And then at this point here, right, let me zoom out a little bit. So the first thing I would do is I would obviously highlight to show the difference between the, not do it that way. Got to do it backwards because I'm doing it on this. So if I'm going annoying tonight, I'm going backwards. There's my GP. And then here is my TP, RUBP. So RUBP, come on. I'm going to be annoying there. So that's the RUBP, and that was my GP. So you should know, if we're now looking at this and thinking about the Calvin cycle, so we know that carbon dioxide comes in, carbon dioxide will combine with RUBP, ribulose biphosphate. So we've got a 5 carbon molecule here. We know we've got a 6 carbon molecule here because we've got a 5 carbon adding in a 1 carbon. That will then automatically lyse itself into the 3 carbons, which is your glycerate 3 phosphate, our GP, and then that will then convert to our TP, and the TP will make its way back to RUBP. So just very basic here on the structure. So you were confused why the RUBP concentration decreases after we reduced carbon dioxide concentration. So we know that RUBP and CO2 are going to produce our hexose biphosphate that doesn't like being a hexose biphosphate, so separates, lyses into those 2 GPs. If I have no CO2, so if I'm reducing the CO2 concentration, that therefore means I'm going to not be able to make any more GP. So therefore, the RUBP would be increasing in its levels. So you're asking about why RUBP concentration decreased after you've got the reduced CO2. Well, the carbon dioxide levels, so you're told here that the carbon dioxide concentration was reduced. The carbon dioxide concentration being reduced at that point, there's still capacity for the RUBP to be converted into the GP. So you've got GPs going down because the GP that was there was converted into TP. The TP that is being converted back up into RUBP is what's causing this rise up. Does that make sense? Do you understand that rising of the RUBP from the fact that we've got our TP being converted? Okay, so you understand that part, but what's not making sense to you is the fact that the RUBP levels are going to decrease. So this is where you need to understand that your GP levels are decreasing. Those GP levels decreasing means that you're not going to have more TP to get back into RUBP. So your RUBP levels are going to decrease because there's no more TP. If we're not making any more GP, we can't make any more TP. That TP can't make any more RUBP. It's like a lag effect in a sense when you're looking at how it interacts with the two together. So your GP decreases, there's less CO2 to go across, so that makes sense. Your RUBP is going to increase and then decrease. Why is it increasing? It's increasing because the TP that was produced before was converted, regenerating the RUBP. But RUBP increases because it's not being converted to GP. The RUP will obviously increase as it's still being produced from the TP, but the RUBP will decrease because there's less GP being regenerated. And so therefore, it's like a knock-on effect. So it's kind of like this cycle going around. It's on my YouTube channel. So, hi, Iris. Does that help at all, Andreas? Does it help in the fact you've got to think about that link from one place to another, that you've got one, because you've got one, therefore means that, oh. So then why is the concentration RUBP not constant? Like what happens after? Okay, so you also, it's where, I don't want to confuse you too much, but you also have to remember you've got photorespiration. RUBP can actually be converted into the alternative substances if I brought in photorespiration. So let me just come away from here 1 second. Let me go back to this. And then what I'll do is I'll bring in the bit about the photorespiration. So it's not just that the RUBP can be used for the Calvin cycle. What you have to remember is that it can also be used in photorespiration. And therefore, it actually, when there's a low concentration of CO2, O2 becomes a factor. It's a competitive inhibitor. 1 second, I'm just trying to bring in where the actual picture is of it. Here we go. Let me bring this in to this part here. So here you have, Rubisco, the enzyme is going to be used to convert RUBP and carbon dioxide to produce your GP, right? So we know that carbon dioxide comes in, Rubisco ribulose biphosphate is going to use RUBP. It's going to fix carbon dioxide, and then we're going to end up with those two GPs. And what we get is oxygen as our waste product, and that is photosynthesis. So then if we look at the fact that oxygen can actually be a competitive inhibitor for Rubisco, the enzyme that would be able to actually that's not the right word I want to use, convert RUBP, fix the oxygen with it, and what you get is a 2-phosphoglycolate. So when those CO2 levels are really low, i.e. they have been reduced, what you now have is a oxygen binding competitively because it is able to fit in that active site. And when it does, we get a new product being made. And that new product is then going to be the 2-phosphoglycolate. That will then produce carbon dioxide. But what you have to understand is that binding and that production of the phosphoglycolate is a toxic substance that needs to be removed. Therefore, energy from ATP is required to convert it to a useful metabolite back to something that can re-enter the Calvin cycle. So, right, okay, well, that's the bit that was missing maybe from your knowledge. When you look at that question, it's expecting you to understand that the RUBP levels will decrease because they're being used somewhere else. They're being converted by something else. And if you look at the answers that they give you, so there's only one last part for that mark scheme where it's asking you about understanding that. If I go back to where am I going? I can't find it now. There we go. This one here. If we go back to here, then there's only one mark, which is to explain this one, which is on the mark scheme as marking 0.6, because actually they are wanting you to really note that it's the fact that the RUBP without carbon dioxide can't be converted into GP. If the GP that was already made is converted to TP, that will then go back to RUBP. So it's expecting you to link those two together, but what it does realize is that some students know and can talk about photorespiration. They don't tend to ask very many questions on that, but if they do, then it's one to note. Okay. Was there any other questions that you weren't so sure? Cellular control, do I need to know physical chemicals and biological emissions and what they do? You need to know the fact that they can speed up the rate of the spontaneous mutations. However, If they're going to ask a question on that, I would suggest that they're going to give it to you in the stem of the question, would be my answer to that one. You're very welcome. If you want to join on I'm actually on my YouTube channel with my slides up. I'm kind of trying to jump between the two sessions. But yeah, Iris, do you have any questions? Andres, is there any other questions that you're not sure about that you want me to look at tonight? Obviously, these sessions are a way of me helping if you've got any questions. They're not planned, so I literally just go with the flow of how things are. You are very welcome. Very welcome. Since we started on the question, let's go. We could look at this one. I quite like this question, actually, this paper, because this paper actually shows you a way in which there is an application to the topic. Perfect. Let's do this question then and see how we can apply our knowledge. So this one, and obviously whenever I do these questions, guys, I never give you just the questions that they have asked in the exam. I'll show you how they can take these questions and take them in a different direction. So what we've got here is we're being told that we have a fluorescent dye. Well, what's important with a fluorescent dye? The key thing is it's going to make things visible. So it makes things visible, so we are able to visualize something, whether that be a practical. So if they were to ask you about a practical, they could ask you to explain how, for example, using the TLC, if you're doing a thin layer chromatography, how is it that you would actually visualize something like amino acids? So in that case, what you'd be doing is you would want to talk about the fact that you want to use anhydrine as your spray. If we're talking about looking at cells in a microscope, what do we want to do? Well, we want to ensure that they are able to be distinguishable. So therefore we do differential staining. So whenever you're looking at these questions, take your time whilst you have the time. I know it's feeling a bit pressurized right now because we're getting close to the exams, but whilst you're practicing, take the time to find some hints to get your brain activated and thinking about how that can move forward. So we have the fluorescent dyes. They are used to measure our concentration of calcium ions. So we know that we've got calcium ions inside the cell. So we know because they've showed us in the question, there is an expectation you know this, but the calcium ions are positively charged. So that's important for us to note. Then they've said, okay, the structure of the Fluor-2 is shown. Again, they give you little hints in the question whenever they do this. So they've given us a picture with the structure, and I'm going to highlight these regions here. And the reason I'm highlighting these regions is because just like I can see up here that calcium is positively charged, what I can also see is that we have got negatively charged parts to this molecule. So this is an important hint in the question to show me that, for example, why it is unable to cross the cell membrane. Well, it's unable to cross the cell membrane because it is polar. Or we could say charged. Or we could say that it's hydrophilic. So we can use any of those terms to get the marking point. So the fact that it is polar, the fact that it's charged, the fact that it's hydrophilic means that I have no idea. I potentially did a question on this. This is the thing. None of my stuff is actually scripted. So I see a question and I do it. And yeah, I probably did actually, because I like this question. And so therefore it shows you that part. And so it can't pass through the phospholipid bilayer. So what are they actually going to ask you? Well, they're never going to give you this question again. So that's an easy one. We can talk about the fact that it's polar, that it's charged, that it's hydrophilic. But what could they do is they could ask you about, well, why can it not get through? What properties of the phospholipid bilayer actually prevents this molecule from getting across. So you stating that it's polar charged hydrophilic doesn't actually explain why it can't get across the cell membrane if they want to test your knowledge on phospholipids. And I do feel like there's going to be quite a lot on lipids coming up, whether that's phospholipids, whether that is just lipids in general and triglycerides, or whether that's linking it all together. That's how I feel that this topic area is going to go. So do you know that the reason it's unable to cross the membrane is because, if we look at it a different light, of this hydrophobic region? And do you understand why this hydrophobic region is hydrophobic? Do you understand why the fatty acids are hydrophobic? Do you know that they have lots of carbons with hydrogens, and therefore, because they have lots of carbons and hydrogens, they have an even distribution of the electrons, and therefore, because of that, they are hydrophobic, and it's anything over 12 carbons makes it more insoluble. So do you know about the insolubility of the lipids? So if you're going to do these types of questions, as I said before, they're never going to use exactly the same questions. What you need to be doing is actually trying to find other topic areas and how it could link together, which is easier said than done. I mean, you've got to remember, I've looked at this specification in and out, and I've looked at all the questions, and I can see where to take this. So I'm not saying this is an easy one to do, but it is one of those questions that I would say definitely do practice on. And then I probably did do this one about the neurons potentially and about the calcium ions, but I don't know. I can't remember. I've done so many videos. I have no idea which ones I have done, haven't done. So basically I can do them over and over again, and each time they're probably slightly different. But we can carry on with this if people would like, or if there's another question that you'd prefer to do, then please do ask away. As I say, these are not done like I do my actual sessions. My sessions are planned. My sessions are very meticulously planned so that I know what I want to show. But I basically build these resources around for these sessions, what whoever's in the chat wants at the time. So if you are here and you want to ask a question, please do, because that's what I want to do. I want to help you with what you don't know. So we can continue with this question, or we can move on if you have already done it. Iris, have you done this question? No, they're not. Yeah, they're out of the spec. So basically, ignore that one. They will not ask this question because it has been removed. Yes, you're absolutely right. It has been removed. So this one, I actually really do like this question. And the reason here we go. We got one who's asking for specimen paper 117C. Right, give me a second. Let me jump to that paper. So specimen paper 1, paper 1, and you want a specimen. So I'm going to have to download that one. I will continue on with this question before I move over to that one. So see if you can answer the question on your own whilst I get this next paper. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66, 67, 68, 69, 70, 71, 72, 73, 74, 75, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, 100, 101, 102, 103, 104, 105, 106, 107, 108, 109, 110, 111, 112, 113, 114, 115, 116, 117, 118, 119, 120, So what do you think? What would you say would be the answer to that one? Anyone guess? Oh, that's okay. It's okay to actually admit that and to say, do you know what? I'm not sure. Because a lot of the time, that's the thing of you do a question, you do a past paper, and you memorize the mark scheme or you use the mark scheme, you read the mark scheme, and then you try and do the question again. You're like, I actually have no idea. And that is a way of identifying that you don't actually know the content. And so therefore, it's always good to review again. So let's look through this and then we'll talk about the next thing. So what are they telling us? Well, they've told us it's a sensory neuron. So the first thing I would be doing is I would be saying, right, I've got a sensory neuron, and I would draw a little diagram here that's been stimulated. Now, they've told me in the question that we're at a synaptic bulb here. So therefore, I change my colors. I'm going to draw my postsynaptic membrane here. So I have my sensory neuron is stimulated electrically. It's got the fluorescence dye, and it's going to be measured within that synaptic bulb. So we're actually measuring it in here. So I'm just going to, let me change color a second, make more sense if I go red again. So in this synaptic bulb, we are going to measure the fluorescent. Then they've told us that an electrode is going to record what happens to my postsynaptic membrane. So what I'm going to do right now is just highlight so I know that this is the fluorescence in the synaptic bulb and that this is telling me about the highest membrane potential in the postsynaptic neuron. So therefore, I need to now use my understanding of what happens when a impulse is actually passed on from one place to another. Guys, if you are wanting to join, I am on my YouTube channel. I'm live streaming this. You can get to the Zoom link if you click on the link and join us. I will try and jump between the two. Right now we're doing 2020's paper. I do have up the specimen paper, and I will look at that in a minute for 17C on that one. But let's just talk about what we've got. Well, we know we've got our membrane potential, which is measured in millivolts, and we should know that our resting potential is minus 70 millivolts. So if we've got minus 70 millivolts, we've got a resting potential, we know that anything that takes us away from that resting potential, we are going to depolarize the membrane. But what's really important is, are we actually depolarizing it and meeting what's known as the threshold value? So have we reached the threshold value? So if our stimulus, whatever that stimulus is, does not reach our threshold value, we will not generate an action potential. So therefore, what we can see here is when there is a low electrical stimulation of our sensory neuron, we find that there is a low fluorescence inside the synaptic bulb, and the highest membrane potential that we get to in the postsynaptic neuron is minus 60 millivolts. So basically we're saying it's getting to about there. It has not yet reached a threshold value. So we know it hasn't reached the threshold value because the highest membrane potential that it gets to is minus 60 millivolts. Whereas we know, because of the all or nothing response, we know that if we have a stimulus that reaches the threshold value, that will trigger an action potential, which means we will depolarize, we will repolarize, we will hyperpolarize back until we get to that resting potential. So therefore, we know our highest point is plus 40. We're told that this one only reaches minus 60 millivolts at its highest postsynaptic membrane. So therefore, we have not got a stimulus which has reached the threshold value and triggered an action potential. Now let's look at the next one. So when we get to a medium stimulation, we get a medium concentration inside the synaptic bulb. We have therefore reached the highest point of plus 40 millivolts. So straight away, I can see we have an action potential here. I can also see that we've got an action potential here. Whether it's high or whether it's medium, we get an action potential because the highest membrane potential is plus 40 millivolts. It has gone up. So then what you then have is, well, what's the difference between them? What's the difference between whether it's medium or whether it's high? Can we really say that there's a difference? Can we be sure? Because it's an all or nothing response. So then if we look at what they've also told us in the question, they've told us about the concentration of calcium ions, and that is determined by the intensity of that fluorescent dye, that it's proportional. So if I've got a medium fluorescence in that synaptic bulb, I can therefore assume that there is a medium concentration of calcium ions. So again, the scientists concluded they love this. So this question is actually module 1. This question is assessing your ability to evaluate the type of information that someone else has concluded. Can you take their results and can you say for certain that what they have stated is correct or not correct in the case? And that's the thing about an evaluate question. The minute you see that evaluate, you always need to give for and against. So try to always give that for and against to be sure that you are hitting the evaluate, and then the explain part of the question. So the second part, explain the scientist's conclusion. How does the concentration of calcium ions in that presynaptic neuron cause an action potential in the postsynaptic membrane? So therefore, do you know that at the synapse, you have calcium that will move in, causing my vesicles, which contain my neurotransmitter, to then fuse with the membrane and therefore releasing the neurotransmitter into the synaptic cleft. Do you understand that if there's a low concentration of calcium ions going into the synaptic bulb, therefore we can assume that not very many neurotransmitters are being released into the synaptic cleft? So it's kind of linking those together to work it all out. Right. Okay, so let's jump. Does that help with that question? What I want to do is, I was asked about 17C on this paper here. So let's see if I can find, where's 17C? 17A, 17B, 17C. Okay, so let's see what they've said on this one. So this is a specimen paper. This is paper 1. So we're looking at 1, 2, 3, and 5. Right. Let's start with just seeing the information they've told us. Right, liver cells have a high metabolic rate. Okay, so we've got hydrogen peroxide. It is a metabolic product produced in significant quantities in liver cells. It needs to be removed because it's toxic. And obviously with the liver, we know that one of its roles is to detoxify. So it needs to do it, preventing damage. Yes, right. So hydrogen peroxide is detoxified by, gotta love an enzyme. Our catalase enzyme. So we know it's an enzyme. So because we know it's an enzyme, straight away what I'd be doing is I'd be saying to myself, okay, we're looking at an enzyme. We need to think about the fact that we've got our active site. We need to consider the fact that it is a globular protein. We need to consider the fact that it has a tertiary structure. And the active site will be complementary. So I'm just giving some words here, complementary. to the substrate. So they've told us we've got hydrogen peroxide, that should be an arrow. That is then converted into water and oxygen. So, yes. So with regards to catalase, it is an intracellular one, that is correct. And they have asked a question in the past about, actually, I think it might be this question, where it's found in peroxisomes. There we go, it is this question. So very high turnover number, right. Catalase has a very high turnover number. So what that's telling you is it works in a way that's going to be efficient. So a single catalase molecule can catalyze the breakdown of approximately 6 million hydrogen peroxide molecules every minute. It's found in these peroxisomes inside the liver, and these are organelles surrounded by a single membrane. Now, this is an interesting one because what we're talking about here is we have got a membrane where these enzymes are being held inside, a bit like immobilization of enzymes. Then if we move on, so they're told us the activity of catalase was investigated in a laboratory using chopped liver tissue and dilute hydrogen peroxide. When the chopped liver was added to the hydrogen peroxide, large quantities froth as oxygen bubbles were produced in the liquid. Shows the effect. So we've got enzyme concentration, we've got the rate of reaction. So as we increase the enzymes, what are we doing? Well, we're increasing the enzyme, therefore we've got more active sites. Therefore, we would expect the rate of reaction to increase because we have more enzymes, more active sites. Therefore, a faster rate of reaction can occur. Identify 2 variables that need to be controlled. Well, this is just, again, assessing your knowledge of module 1. How do you do practicals? So do you understand that things like, and I'm just going to write this up here, variables, you should have a go-to list. So have they controlled? Absolutely. Have they controlled the pH? Did they tell you that they put this in a pH buffer? Have they controlled the pH? So if it goes on to say, oh, if it goes on to say exactly as it does, how could you control the pH? That's the sort of thing they're looking for you to actually tell them. They want you to give them the information that you know that the controlling is necessary when you are looking at the rate. You need to control things like pH. How are you going to do that? Well, you'd use a buffer. So do make sure whenever you are doing these questions that you have your go-to answers of how to improve, how to make things better. So pH is absolutely one, and how would we improve it? We'd use a pH buffer. What else could we do? Well, actually, maybe we can't use a buffer. So instead, we could, so either using a buffer or we could use a pH probe to monitor. So don't think that it's just a case of you have to control something. You can also give them the information of ways in which you would monitor that so that you can then account for any deviation that occurs based on that. Hi, guys, if you want, I am on my YouTube channel. I'm doing this and on the Zoom, but obviously I'm kind of jumping between the two. Is there any other control or any other variables we need to think about? Well, they've told us the concentration of the enzyme, but did they tell us how much of the substrate they put in? Because concentration isn't just about the enzyme, you need to control the other parts of it. So the concentration of whatever else there is, so concentration of the substrate would be 1. Did they tell you about the volumes of what they were using? Did they tell you about the temperature that they were using? So if they haven't given you any of that information, then they'll be looking for you to tell them how you would control those bits of information. So how would you control the temperature? i.e. potentially put the conical flask into a water bath. How could you make sure that this was correct? Well, you would ensure that they are going to be set at a specific temperature and have a thermometer in there to check it. So if they give you, oh, is it not? Hold on one second. Where's it gone? Oh, do you know why? I bet it's not working because I've actually closed down the thing that I had open. 1 second. That was me jumping around too much. Sorry, guys, give me a second if I go to my channel. And then, oh, yeah, there's a whole load of people here. Sorry, guys. I did not know that that completely cut you out. Right, give me 1 second. Let me just put the invite in here. Sorry, Paul, thank you for that. You did say before, but I wasn't paying attention. Sorry, guys. So to answer the question with regards to, do I think PMT predicted papers are worth doing? Any practice that you can have is worth doing. So just to be clear, any practice you can have is worth doing. Do I think the predicted papers are the best way to go? I don't know, because I've made predictions of what I think could potentially come up. But as I've said in the past, as I'm always saying, I might get it right, I might get it wrong. I don't know. So it's good to do it, but not make the assumption that it's definitely going to be on that paper. So that's one thing I would say, to try your best to not assume that it definitely does mean that you are going to only need to study that topic area. So make sure you're studying everything. But any type of predicted papers is basically someone like me sitting down and going like, well, what could come up? How could I plan some questions around that topic area? But unless you're the examiner, you don't know. But I definitely do think papers are a good practice for you, but just make sure you are revising. Right. Okay, stop playing my hair now. Okay. So with regards to any type of practical question, I would say that making sure you are looking at having a list of go-to answers so that you can say, right, these are my go-to answers, and now I'm going to apply it to this situation. So I don't want to say things like mass, because actually, am I controlling the mass? Well, no, they chopped the liver up. So they chopped the liver here. They've used chopped liver. So have they controlled the mass of that chopped liver? Well, no, they haven't. Could I use that as an answer? It depends. If I'm going to use the mass, then I do need to be specific in the fact that why would that matter? It's chopped up. So therefore, I haven't controlled the surface area of the exposure. So it's sometimes they will allow those types of answers and sometimes they won't. And it all comes down to the examiners that are marking your papers. And this is why a lot of you tend to say, why is it that one mark scheme says it and another says something different? And the reason being is because different years, different examiners will come together and agree whether or not they will accept the answer or not. Anita, do I do AQA too? I tutor AQA privately, but I don't do resources for AQA yet. I'm not sure if I will do AQA. There's so much out there already. What I realized this year, well, yeah, technically last year, but when it came to my students, I didn't have anywhere to direct them for OCRA. And so that's why I built my YouTube channel. But obviously there's Miss Eastrick, there's BioRach, there's like so many people out there, but I couldn't see anyone who was a specific teacher for OCR who'd built resources, which was why I made what I did. Do I think the N14 and N15 is proof the semi-conservative replication model will come up? I don't think it'll be as straightforward as the question they've done in the past. I do think that DNA replication and linking it to mutations and then linking it to evolution would be a way in which they are assessing those together. I feel like there's definitely going to be a lot more on module 6 to do with patterns of inheritance, but potentially looking at the what am I trying to say here? Looking at the points where it's maybe epistasis, maybe there's a mutation that results in, for example, the production of a repressor protein. I do think regulations are coming up. I definitely do feel that that's on its way. So yeah, I hope that is helpful. Did that answer your question? I'm not sure if you are still here. No, it's not 175 for 1 hour. It's 7 weeks. It's 7 weeks of 1 hour group revision. So you get oh, yeah, 175 would be mad. Absolutely. No, it's not what yeah, it's 7 weeks, so it actually works out at £25 an hour. So it's the cheapest I could get it for you. And it is limited on the number of people who can book in the course. That's really funny. Right. And it was, oh, hold on. I think you actually said 3, didn't you? And I've just gone on the ones before. Is that what you said? Where has it gone? It was 3C, yes. So now it says using this information to deduce why and how catalase activity is regulated inside the liver cell. Well, this is where we have to go back up and note what they've told us, that these are in a peroxisome. So the fact that they've told us in the question that these are found in peroxisomes. So they've told us they're found in peroxisomes. They've told us that a peroxisome is an organelle surrounded by a single membrane. That is telling us that they are actually being contained in a membrane, so they're not functioning all the time exactly as they would do if they were free floating within the cytoplasm. So what they're looking for you to understand with your reasoning on this one is that you're producing a large quantity of these. They told you it's a very high turnover number. That high turnover number is going to be producing a breaking down the hydrogen peroxide into those oxygen and water molecules. So you're going to have this large quantity of hydrogen peroxide. You're going to have a high turnover number of your catalase, and therefore that's going to be a really vigorous reaction. That's a lot of energy that's going to be produced as well. That oxygen production very quickly. So in a way in which they can enable this to be, as it says here, why and how the catalase activity is regulated. How is it regulated? Well, it's regulated because it's held inside the peroxisomes. Therefore, if it's held inside the peroxisomes, it's not going to be released in the same quantity as what it's being made. So it's being kept basically in a cupboard until it is needed. So you can either think about the fact that you are isolating the enzyme, so it's only able, a bit like if you think of your phagocytes, and your phagocytes have your lysosomes, and your lysosomes contain your lysozymes. The lysozymes are being held within the vesicle until they are needed and required. So in this case, this is the way in which the liver cells are isolating that catalase in the peroxisomes. But remember what I said before, catalase is an enzyme. What did we say about enzymes? Going back up, enzymes are globular proteins. Globular proteins which are produced through transcription and translation. And remember what I said before, transcription translation actually is a process which can be regulated if we look at the regulation of gene expression. So therefore, the cell might actually limit the expression of this enzyme until there is a environmental factor that signals that that should be expressed. So therefore, we could limit the concentration of the enzymes. Therefore, going back to the graph, if we're using less enzymes being produced, we're therefore reducing the rate and therefore it not causing a problem. Does that help? Has that answered the question on how to link those ones together? This one is, on this last one, is a 6 marker. This is basically what you think you can answer, but 2 parts to it. You need to give a why, you need to give a how. Why is it needing to be regulated? And they gave you the hint there, regulated. To regulate that activity, the activity of the catalase is to do with the environment as well. So there's always going to be little hints in your questions. You've just got to find them and see how that would come together. Right, does anyone have any other questions they want to cover? Or we could carry on with this one. I don't really feel like photosynthesis is coming up that much. I do feel like ECG is going to come up, and I feel like ECG is going to be linked with the animal response topic area and the neuronal communication. I feel like those are going to be interlinked somehow. Whether it's on, you're very welcome, whether it is on module in paper 1 or paper 3, it is definitely going to be a linking of neuronal communication. And I do think that muscles and cardiac muscle might come together. So although you have learned about skeletal muscle with regards to the sliding filament theory and the sarcomeres and the shortening, remember we're talking about muscles. So actually, when you talk about muscles contracting, you should know that for the heart to beat, for the ventricles to contract, for you to have ventricular systole, that contraction that's occurring, that is the sarcomere shortening. No, H, to answer your question, would I do a video on my predictions? I don't like to do that because I basically, if you ask me on a different day, I'll tell you different things based on what thoughts have popped into my head at that time. So I definitely feel like muscles, I definitely feel like neuronal communication is coming up. And I feel like they'll do it in they'll start off, for example, with the skeletal muscles and the sarcomeres, but then they'll move on to, for example, the cardiac system and looking at the neurons involved potentially in the cardiac system and how the sympathetic or parasympathetic will link. But if you ask me on Wednesday, when I do a session again, I might come up with a different prediction. I do feel lipids is coming up as well quite a lot. But again, it just depends which way they want to go. There's so many different ways that they could answer or ask the questions that it's really just dependent on who's written it. That's the one thing I would say to you. It's not as simple on as just me predicting and getting it right. I have gone through every single paper. I have gone through every single question. I have looked at every single topic area and I have analyzed them, but I don't know. Yes, Andreas, I do feel like coordination of the nerves and endocrine system could come up. I feel like it's one of those ones where I definitely feel like neuronal system, absolutely. They have not done much on neuronal and the muscles. I would definitely say that they are potentially going to come up. Lynn, I have seen your question. Don't worry, I will cover that in a second. But it's just how they do it. That's the thing that I would say. Oh no, I don't want to do that. Give me 1 second. So the Zoom link, let me just share that again. 1 second. There you go. Right. Yes, Mary, give me a second. Let me just answer the question on the RQ, and then I will move on to that. So if we're looking at respiratory quotient, then if they're going to ask a question on that, they are looking for you to note. Let me just bring up a quick thing on this. This one. So I'm just going to do a quick whiz through on this. So they're expecting you to note the fact that, no, went too far. You should know respiration. You've got glycolysis, you've got the Lynch reaction, you've got the Krebs cycle going to oxidative phosphorylation. So that's respiration. We know respiration. They asked a lot of respiration last year. Does not mean they won't ask about respiration again. It just means that they will ask it in a different way. So the respiratory quotient then is basically where we're looking at how much CO2 has been produced and how much O2 has been consumed. So they did ask a question on this last year, so I don't feel like they'll ask a big one, but it would potentially be a multiple choice question. So you've got your carbohydrates, glucose, 6 divided by 6, so 6 oxygen are consumed, 6 carbon dioxide are produced, so 6 divided by 6 gives you 1. Now, if there isn't carbohydrates, if the respiratory substrate of choice isn't available, then what can happen, and this is where I think they may go, is where the lipids can be broken down into glycerols and fatty acids. Glycerols become pyruvate, fatty acid becomes acetyl coenzyme A. Therefore, this time we're needing 25 oxygen, but only 18 carbon dioxide molecules that are being produced. Therefore, the respiratory substrate, the respiratory quotient is 0.72. If we're looking at proteins, it's, for example, in this case, 0.6, 0.54. So 0.54 of carbon dioxide being produced, 0.60 of the oxygen being consumed, therefore the number is 0.9. So if you understand that, therefore you understand how they can actually enter and where they can enter, then they could tell you using a respirometer, or they could ask you to plan an investigation on this one. What's your independent variable? What's your dependent variable? What are your control variables? So they would give you, for example, 37 carbon dioxide produced, 46 oxygen consumed, then expect you to do the calculations. They won't give it to you in the equation, but they will expect you to know which way around to put the numbers if they're going to do that one. So that is, excuse me, how they would do a respiratory quotient if it was a multiple choice question. Okay, so let me just look at the biotechnology part first. So, Mary, when you say biotechnology, where are you wanting to go with that? Are you wanting to look at a question example, or do you just want to know the content of that? Content, okay. So if we here's one I prepared earlier. And this is what I basically do. Right, which links, and it means I can bring in the other part as well. Okay, so biotechnology, oh, that was just me saying basically protein stores, carbohydrate stores, lipid stores, because I should know my numbers of 0.91, 0.7 as a conclusion. No. So there is an expectation that you know the number 0.91, 0.7. That's an expectation, you know that. Then it's an application. So last year's paper, they asked it where they looked at, you saw the numbers. Hold on, I'm getting these questions because they've used the question before. So one was about wattage and one was about exercise and one was about the types of exercise. So it's more about applying the knowledge. If they're going to ask about why one value would be higher than the other, then they'd literally just be wanting you to talk about how many carbon dioxide produced, how many oxygen. If, however, now what you've done is you've piqued my interest and you've now made me go, ooh, well, they could ask you with regards to why the 0.72 is lower with the lipids, and understanding the fact that your fatty acids can be broken down into many chains. Or they may ask you about the fact that if you have your what am I trying to say here? Anaerobic respiration. The anaerobic respiration would give you a higher number than 1. So they have given a mark for that part of the content before, but that was on a mark scheme because I see what I need to do. Hold on 1 second. Yeah. It was on the mark scheme where they wanted you to explain that above a certain point, you were then doing anaerobic respiration, but they'll never ask you to go into more detail that is more complicated than what it needs to be for your exam, if that makes sense. So yeah, I don't feel like they will ask that question. However, I may be wrong. This is the one thing. Right, let's have a look at biotechnology then. So one of the key things you do need to know about logarithmic scales. So they will ask you about logarithmic scales. It seems to come up. It's come up 3 times now in the last 3 years, and it does seem to be a common theme that they're asking more and more. Now, whether or not they ask you to... Whether or not they're asking you to draw the graph, or whether it's I feel like you're going to either have a line graph or a histogram to draw this year, because it was a bar chart last year. So to answer your question on that one, you're looking at how much oxygen needs to be consumed and how much carbon dioxide is going to be produced. Well, when you've got glucose coming in, glucose, you know, is 6 carbons to 6 oxygens. So the carbon to oxygen ratio in a glucose molecule is 1 to 1. Whereas when you look at a fatty acid, the carbon to oxygen ratio is much lower. And so because of that, what you've got is the fatty acid chain is coming in at this point here. So the fatty acid chain can only come in at the acetyl group. Therefore, the carbon dioxide that are being released are only being released here in the Krebs cycle. You don't have your link reaction decarboxylation occurring. So that's why the carbon dioxide produced number is less when you're looking at the breakdown of a lipid compared to the breakdown of a carbohydrate. So it's to do with the ratio of your carbons to oxygens on that front. Hope that helps. Right, logarithmic scales used as a number. And what you can see here is typical growth curve. You've got your lag phase. No bacteria dividing, cells are getting used to their environment. We've moved them from one place to another. They are starting to make enzymes to deal with the food source that you have provided them with. Now remember, so this is where I do feel like you could link things together here. With, hold on 1 second, sorry. You can link things together here with the enzymes. So I want to just bring this up. Making enzymes. Well, what are we talking about when we're talking about making enzymes? Well, we're talking about transcription, translation. What else are we talking about here? We're talking about their environment and them getting used to the environment. Well, this is an interesting thing because actually this could link to gene regulation and the fact that the environment is going to regulate whether or not the genes will be expressed. So if we're looking at, for example, bacteria and dividing, we could be looking at the lactoperon if the food substance that's being provided to them were lactose, for example. So I'm just throwing something out there. I've not seen questions or anything like that, but I'm just showing you where it could go. And that's the thing you need to know, is that they could take it any which way they want. They've given you some information, or in this case, I have. And all I'm doing is I'm saying, well, what could I assess my students on here? Well, I could assess them on, do they understand how the environment will have an impact on the organism? Well, what does that mean? The environment having an impact on whether transcription or translation is occurring. So if I provide lactose, am I therefore going to stop the repressor protein from binding to the operator? And if I'm stopping it binding to the operator, then RNA polymerase can now bind to the promoter region, and those three genes, those structural genes, can be transcribed and then be translated later on. So if I want to, I could ask that question, potentially. Or maybe I want to check and see, do they understand that lactose is a disaccharide, and that disaccharide is held together by glycosidic bonds between a glucose and a galactose? Or I could say, well, do they know that lactose is actually a reducing sugar? So do they know the reducing sugar test that could be used to test the food source that's been provided? Again, just going off on random tangents here. But all of it's interlinked. If you understand your fundamentals of module 2, then they can throw that in at any point throughout the paper. So don't think that they're only going to give you it based on this topic area. So it's an interesting one, Paul. What they do with OCR is they definitely do it in the sense of they expect the students to have the bigger picture and to link together, and they will ask the questions to direct that linking. But if the students aren't able to see the bigger picture, they lose marks because they can't see where to go. I was talking about this earlier today, and the fact that AQA, a lot of people who say, do you do AQA and what do you think? So AQA have easier short questions, but they expect their students to do that long 25 marker at the end and look at that logical linking together in paper 3. So they have to do it as an essay style, but they still have to do that linking and application points. Whereas with OCR, they don't have the 25 marker. What they have is they have short questions that they want them to then add the information to. But the thing is, with OCR, they give them hints throughout the question if the student knows how to find them. So they do more on application skills than just the writing an essay. And that is the bit that becomes harder. But then some students don't do well in writing essays. So it's a way of deciding which exam board that you want to use based on how you think your students would actually do better in the exam. I have to say, of all the exams, Edexcel B, I'd say, is a horrible one for how they assess the students. Do I have a list? No, I've already covered that because I don't believe in predictions, although I will, on these sessions, think about what pops into my head and say it. So if you come to any of my live sessions, and they are on my YouTube channel, then you'll see what things I come up with. I do have thoughts and I do air them, but I don't want to put it out there in case I'm wrong. I have a moral part of me that's saying don't predict, because I don't want to make students believe that I know something that I don't know. It is just a general, my guesstimation, basically. Right, back to this log phase. Bacteria dividing at a constant rate. So if they're dividing at a constant rate, how are they dividing? Well, this is binary fission. So binary fission is what's happening here. And then you have your stationary phase. Your cells are dying at the same rate as them being reproducing. So therefore, we have this plateau. Now, be aware, if they do give this to you as a question in the exam, they will expect you to actually note that the point where, for example, that stationary phase may happen. So if we were doing batch fermentation, for example, where's the time period for us to actually be removing the products so that we don't get to that death phase? So we're talking about the carbon and hydrogen bonds. I was talking about the ratio between the carbons and hydrogens compared to the ratio of the carbons and oxygen. The people keep saying this wrong. I've obviously clicked this wrong. 7 weeks. It's 7 weeks for that price. It's not 1 hour. It looks like it is because of where I'm sitting. It's not 1 hour, £175. It's £25 an hour. It is 7 weeks of the sessions. If I can, I will. I'm running out of time. I've literally got 12 minutes left because I need to go to bed. I will be back on Wednesday though, so potentially, although I would say Chi-squared came up last year. Do I think do I think it's going to come up? I don't feel like it is, but then they sometimes throw curveballs in at you and do it again just to double check. Because they think people are like, oh, well, it's not going to come up again. So they don't memorize or learn that part. If you haven't, check out my workbook, or not workbook, study guide. I actually have a study guide, which is on the practical module 1 on my website that you can use, and it goes through with worked examples for all the skills of module 1 on that one. Anyway, back to this death phase. Cells are dying at a faster rate. What have we got? We've got limiting factors. So this is where they could show you some information and ask you about which limiting factor was actually causing that rate to decrease. So the biotechnology side of things then is basically where we're going to apply a biological organism. It could be fungi, i.e. yeast. Now yeast and anaerobic respiration, fermentation, that is one way, because they did just respiration last year, one way where they could link these two together in paper 3. So if they're going to go down the route of biotechnology and want to assess your knowledge on respiration as part of the biotechnology topic area, they will throw in different respiratory substrates. They will throw in a graph which has different oxygen levels, etc. So the 20, it's either 17 or 19 paper has an amazing question on that I would suggest you have a look at if you haven't done so already. Well, it's a bit of a mix and match with that one. They give them some questions, they don't give them some equations, they don't give them others. So there's an expectation that they know their GCSE math skills, like how to measure area, how to measure things like that. But there are equations that are given to them are like the chi-squared, Spearman's rank, t-test, the Simpson's index of diversity is given to them, the standard deviation is given to them, Hardy-Weinberg is given to them. Can't think of any others, but yeah, off the top of my head, they're the ones that are given. There are others that there is an expectation. Okay, so microorganism life cycle, very short. Therefore, they don't require very many nutrients, but we get rapid growth, etc. Genetically manipulation. So again, this is where they could take the biotechnology topic area and link it with your manipulation genome and linking it to how you can genetically modify an organism to produce the required product that you want. So this is a formula that you need to know. I want to say this is one you have to know and recall, not that it's provided with you. But now you've asked me that, Paula, it's like, the top of my head, I don't actually remember. I know I've got the wrong one. Hold on, let me double check. See, this is where I would always have the specification open so that I can double check what do I need to know against what I should know. So I would go straight here. Oh, let me just go away. I don't want that. What was I looking at there? Population. It's like 60. Here we go. So Hardy-Weinberg, Simpson index is provided. Surface area and volume of a cylinder and sphere is provided. Chi-squared, t-test, both t-tests. Spearman's rank, standard deviation is provided. Where is it? There we go. Microorganism population. This one is a recall. You need to know this one and you need to be able to recall it. So basically, you should know that the N, the final number of organisms, is the initial number times 2, which signifies each organism doubles the population size, to the number of generations. So this is the type of question they would give you. In the optimum conditions of a bacterium may divide once every 30 minutes. If the colony was a single cell, calculate the number of cells that would be produced after 6 hours. So they've told you every 30 minutes, they've told you it's a single cell, and therefore 6 hours, 30 times 2, 6 times 2 is 12. So therefore n equals 1 times 2 to the power of 12, which would give you the number. So then they would potentially show it to you that you have to answer that question in standard form, maybe. It could be that they just want the number. So it's important that you're looking at what the question asks and what they've told you. And then they could say, glucose levels have decreased. Why have the glucose level decreased? Well, the glucose levels have decreased because this was the respiratory substrate of choice. Therefore, they were doing respiration. And then this one with the increase in biomass. So Amna, to answer your question, why do you replace bacteria in continuous fermentation if nutrients are constantly supplied? So although we're saying that they're not reaching the death phase, you are going to be having an effect on the numbers that are there. So it's a case of all organisms can't live forever. So you would need to replace them, even though you're supplying them with the constant nutrients, there will be death. The death phase is where the rate of death is greater than the rate at which they are being able to replicate in. So that's why you need to supply more for that ability for them to go through the binary fission. Right, so glucose levels. So the formula that you need to know was N equals N0, which is your starting number. So initial number of organisms, N0, times 2 to the power of N, which is the number of generations. But Sarah, if you go to my YouTube channel, I am live right now, but this is being recorded on the YouTube channel, so you can watch it back. I'm probably, how long am I even in this? I don't know. How long have I been going? About an hour and a half, maybe. Oh, no, cancel that. Almost close you down. An hour and 19 minutes. So you want to look at about an hour and 19 minutes to get to the bit on my YouTube channel as to where I've done. Okay, so glucose levels drop down. Why? Glucose, respiratory substrate of choice. Therefore, this one is an interesting one because they may change this. They may say it's maltose. What would you expect? So they may give you a graph that says that's the glucose. What would you expect for maltose? So they would therefore expect you to know that maltose is obviously made from 2 glucose molecules. And those 2 glucose molecules are going to be hydrolyzed down. And because they need to be hydrolyzed down, that therefore means that the microorganisms need to produce the enzymes necessary for doing that hydrolysis. For example, wouldn't it decline if there isn't glucose available? C, right, that's an interesting one. Where is it the biomass, what is biomass? So biomass is obviously, we're talking about the mass of the organism removing the water. So if we are looking at the biomass and that level increasing, the reason it plateaus and doesn't decline is because these microorganisms are able to produce enzymes that can convert these molecules into other substances that make them more useful. So if you are looking at, for example, let me just move on a second. I want to show sourdough starter. So yeast, flour, and water are placed in a container. So my partner loves making things and had this whole project where he was making lots of sourdough. So this is a sourdough starter. Now, sourdough starters, getting started is easy, but keeping it is actually really tough because you have to constantly be providing them with the necessary substances that they require. So what you have to understand is to make sourdough, what makes sourdough so nice is that it's not just the yeast, the flour, and the water, it's the fact that you have bacteria in there that can actually produce lactic acid. And that's what's actually going to give flavor and texture to the bread and the dough as it's starting to ferment. So this is an interesting one. How are they able to make this lactic acid? Well, they're lactic acid bacteria, so therefore they produce enzymes that are going to be able to convert pyruvate into lactic acid, because we know yeast, when yeast do alcoholic fermentation, they produce ethanol and then ethanol because they produce the enzymes that allow for the decarboxylation and then the dehydrogenation, because they produce those enzymes that are required for that process. Whereas if you look at the lactic acid bacteria, they are going to be producing different enzymes. Ooh, hold on a second. What are enzymes again? Enzymes are proteins, globular proteins, which have a tertiary structure. What determined the tertiary structure? The tertiary structure was determined by the amino acid sequence, which was their primary structure. The primary structure was through the process of transcription translation from the gene that could potentially have had a random mutation that occurred. So this is where they could interlink those different topic areas together. Now, my phone has just gone grayscale on me, which tells me it's now 10 o'clock at night. So I am going to stop there, guys, because I need to go to bed. I will be back on Wednesday again to do a bit more. So guys, thank you for joining me. Obviously I do these sessions as much as I can to help you. If you are interested in attending a proper planned session where I've gone through all the process of putting things together and helping you, then please do consider my 7-week pre-exam prep that I'm doing on a Tuesday evening. But if not, obviously I will see you on Monday.